Okay, hello class. This is Dr. Lyons again, and in this lecture video, we're going to be getting into chapter 9. So that's the genetics chapter. And this chapter is a pretty big one, and it's really a pretty important chapter too, which is why I'm splitting it in half. So we'll talk about the first part of the genetics chapter in this video lecture, and then I'll go into the second part in the second video. So I don't have any pictures of genes, but I do have a picture of this cute little thing which is known as a nudibranch. So this is a small spineless animal that you can find on coral reef areas. And this is maybe one of the cuter ones because it looks like, you know, a little blob with Oreos stuck to it. So before we really get into genetics, I want to just give maybe kind of like a brief overview. So I want to talk a little bit about inheritance and what gets inherited, what are genetics, why do we care about them. So first of all, just briefly, what is inheritance? So we think of we often think of inheritance in the format of things that we get from our parents when they pass away, right? So you might inherit, you know, some money or a house, or if you're unlucky, you might inherit your parents' debts. You know, these are things that we get from our parents. So to inherit something is to get something from your parent, essentially. So obviously, we're not talking about debt or getting houses from your parents or whatnot. What we're going to be talking about being inherited here is DNA. Because as we've been talking about now for a few chapters, DNA gets passed on from parent to offspring, right? So we inherit that from our parents. And genetics is then kind of just making sense of all that. You know, because you get a bunch of DNA from your mom and you get a bunch of DNA from your dad, and we kind of put all this DNA together, and it just kind of magically or mysteriously makes you who you are. You know, it affects, you know, how tall you are and what your skin tone is and you know, and whether you're, you know, whether you have like curly hair or straight hair, right? So all this DNA gets put together and then it just, you know, magically makes you who you are. And so figuring out, you know, how that actually happens, going from the DNA that you got from your parents to what you actually look like, that is essentially genetics. And so why genetics are important then is that it really tells us a lot about who we are. So the two genetics chapters are really a very very personal chapters because they tell us a lot about who we are and why we are the way we are. So that's really why genetics are so important because it has a big effect on us and what we look like and how we behave and such. So let's get a little bit more into genetics and heredity. So heredity is essentially transmitting traits from one generation to the next. So transmitting DNA from one, from one to the next. And genetics is essentially the study of that, right? So it's it's kind of figuring out how all of that DNA kind of works together to make you who you are. And of course, you know, I think most of you know that we look like our parents because of our heredity, because we inherit DNA from them. And what we actually get from them is chromosomes, right? So this is a picture that you saw before, right? So this is the 46 chromosomes, or most humans have 46 chromosomes, so 23 from mom and 23 from dad, right? So we get that from our parents. So there's some from dad and some from mom. So genetics and heredity are what link together two important things that we're going to be kind of talking about throughout this chapter. So it links together genotypes and phenotypes. So genotype, that refers to the actual letters of DNA that a person has, right? So here's a cartoon picture of DNA, which, as you know, is not actually what DNA looks like, but it does come in that general shape of the double helix. So the genotype is the actual DNA, the actual genes that somebody has. Whereas then the phenotype is the actual physical appearance of a person or of any living thing for that matter. whether you are, say, tall or short, right? That is phenotype. That's the actual physical appearance, the physical manifestation of the genes that you have inside of you. So this blue arrow here, this is essentially what we're going to be talking about in this chapter, right? How we get from the DNA that is inside of a person or any living thing for that matter, how that then affects how that person looks, whether they're tall or short, or or balding or not balding, those sorts of things. And so any discussion of genetics pretty much always is going to start with this guy. So this guy his name is Gregor Mendel, and he is considered the kind of grandfather of genetics. So Gregor Mendel, he did some experiments back in the 1860s on pea plants. And so he did some very simple experiments just in his garden, growing pea plants and taking the seeds that resulted from pea plants and then growing them and seeing what they look like. So he's just doing some very basic experiments, just reproducing pea plants with each other and seeing what happened essentially. And so he's credited with creating pea soup, believe it or not. No, sorry, that's not a real fact. That's a fake fact. But I would have to assume that Gregor Mendel probably had a very good recipe for pea soup, because he was doing lots and lots of experiments on pea plants for years and years and years. So I can only assume that he really, really loved pea soup. And even if he's not credited with creating pea soup, he is a really, really important figure in biology. And he is perhaps one of the more brilliant minds of biology in the past few hundred years. And why I personally think he must have been incredibly brilliant is the timing of all of this. So he was doing his research in the 1860s, right? So research on genetics in the 1860s. But some of you will remember back from chapter 10, we learned that it wasn't until the late 1800s that we actually realized that the DNA was even a thing. And it wasn't until the early 1950s that it was determined that DNA is what is passed on from parent to offspring, and why parents look like their offspring. So he was doing his research almost 100 years before we even knew that DNA was something that gets passed on from parent to offspring. So, but even without the benefit of knowing what DNA is, Gregor Mendel was able to come up with a lot of how genetics works. He was able to essentially learn about genetics without having the benefit of knowing what a gene is, what DNA is. So he was a pretty brilliant. And so the work that Mendel did was essentially taking plants, reproducing them with each other, and seeing what their offspring look like. So in the case of pea plants, he would look at different characteristics, right? So a characteristic might be something like flower color, or the color of the pod, or whether the plant is tall or short. And those characteristics can come in different versions, right? There's varieties for each one of those characteristics. So for instance, with the characteristic flower color, a pea plant can either be purple or white. Or in the case of the color of the seed pod, right? With that characteristic, you can either have a yellow trait or a green trait. So he was looking at these characteristics and these traits. And what he essentially was doing was cross-pollination. So he was taking the pollen from one plant, right? So that's like the sperm of one plant. And he would then brush some of that pollen onto the stamen, or he would brush the stamen, which has the pollen on it, and then he would brush that onto the carpel of a plant, which is like the female part of a plant. So essentially, he could control what's going on here. He could say, okay, you two pea plants, I'm going to make the two of you reproduce with each other. And then essentially he would put a bag over the top of this female plant just to make sure that no other pollen got into there. So he essentially was controlling who was doing the deed with who, so which plant was reproducing with which. And then he would grow up, so then once this plant is pollinated, it would make a pea pod, and then he would take those pea seeds and he would grow them up and then see what they look like. having known what the parents looked like. So this is it was a pretty ingenious thing to use pea plants because, they're very easy to grow. It's very easy to take offspring and then grow them and see what they look like. And pea plants don't have feelings, right? So something like this could obviously not be done with humans, right? It would be hugely, hugely unethical to take two humans and say, okay, the two of you, you're going to go, do the deed and have baby, and then 9 months from now, tell us what the baby looks like. Obviously you can't do that. That's hugely unethical. But the things with pea plants is that they don't have feelings, or at least as far as we know, they don't have feelings. So as far as we know, these pea plants didn't mind that they were forced to reproduce with each other. So that's what Mendel was doing. These are the characteristics and traits that he was looking at, right? So we looked at flower color, flower position, the seed color, seed shape, pod color, how tall the plant was. So he was looking at all of these different characteristics and traits, which just kind of gets to a point I made earlier, the fact that he must have really loved pea soup because he was growing crap tons of pea plants in his garden. I kind of imagine he probably had some friends that would always, always joke about him about how he just loved pea plants and just wouldn't grow anything else. I imagine his other friends in the monastery where he worked, because he was a monk. I imagine the other monk buddies coming to him and saying, what's with all the pea plants? Why don't you grow a pepper for us instead of pea plants? Because we're sick of your pea soup. So one really important type of experiment that Gregor Mendel did is what's known as a monohybrid cross. So let me explain what that is. What he essentially did was he would produce purebred varieties of pea plants. And this is in the same way that you might produce a purebred variety of a dog, for instance. So a purebred golden retriever has golden retriever parents, golden retriever grandparents, golden retriever great-grandparents, and then golden retriever great-great-great-grandparents, going back a long ways. So that's what a purebred is. It's something that's the result of mating things that are similar to each other over time. And so he did this with his pea plants, where he would take, purple flowered pea plants and reproduce with them with each other, and then take those offspring and reproduce them with other purple flowered plants, and so on and so forth. So he's creating these purebred varieties. And he would use them in a monohybrid cross, where you take two different purebred plants that are only different on one characteristic, and then reproduce them with each other and see what happens. So if we break this word down, so mono means one, and hybrid means like two things kind of put together, right? Like a hybrid car, like a Toyota Prius, for instance, is part electric and part gasoline. So hybrid is a mix of two different things. So mono meaning one, hybrid meaning mix of two different things. We're crossing two purebred plants that are identical to each other with except for one characteristic. So take, say, a purple flowered plant and a white flowered plant. Those two plants are identical in every other way except that their flower color is different. So that is a monohybrid cross. And then the offspring that would result are essentially hybrids, right? Because they are a mix of different types of purebreds. We often also refer to hybrids in the context of something that comes from two different species. So for instance, a mule is a hybrid. A mule is the offspring of a horse and a donkey. A liger is another type of hybrid. So that is a mix between a lion and a tiger. And a tigon is the same. Again, it's the mix of a lion and a tiger. The difference between ligers and tigons is that ligers have a dad that is a lion and a mom that is a tiger. Whereas with tigons, it's the reverse. So it's a dad that is a tiger and a mom that is a lion. So that's how those two things are different. So what Mendel did was he had his purebred plants. So we've got a purebred purple plant and a purebred white plant. So he made those two plants have sex with each other, and then he looks to see what do the offspring look like. So I want you to think just for a second, pause the video if you want, think to yourself, what might happen? What could possibly occur here? Like, what would the offspring look like? So some potential reasonable, guesses might be, oh, you might have, some purple offspring and some white offspring. Or maybe you have offspring that are all kind of a mix, like kind of a lavender, somewhere between purple and white. Or maybe you have all purple offspring, or maybe you have all white offspring, or maybe you have purple and white striped offspring, right? There's a whole bunch of different possibilities that could happen here. But what actually happened in this particular circumstance is that all of the offspring were purple, right? So 100% of the offspring ended up being purple. So something interesting went on there where somehow the white flower, that white trait somehow went away. And if it's in this plant right here, it must be masked because we don't see it because all of the offspring here are purple. So then the next thing that he did was he took some of these plants and reproduced them with each other. And so probably a lot of you are thinking, ew, that's incest. These plants down here, they're siblings. But again, they're pea plants. They don't have feelings as far as we know. So as far as we know, they don't know what incest is. So anyway, he would cross two of these plants together, so two of these purple plants together. And what do you think he might have found? Right. So again, think, pause if you have to, and think about what might occur here. What's a possibility here? So what he actually found in this circumstance is that on average, if you had four seeds that came out of a pod, essentially three of them would be purple and one of them would be white. So you have a mix of purple offspring and white offspring. And it conforms to roughly a 3-to-1 ratio, where for every four plants, on average, three will be purple and one will be white. Right, so this is kind of a weird result because we have this weird ratio that is kind of unique, and we have these white flowers have come back, right? So white disappeared here, but somehow it came back right here, right? So it's a pretty puzzling result. But Gregor Mendel was a clever enough person to kind of figure out what was going on here. And so he laid forth a couple of different hypotheses in order to explain what was going on with this situation. So the first hypothesis that he came up with is that with particular characteristics, there are alternative versions of genes, or what we now know to be genes, what he called factors back then because he didn't know what a gene was. These alternate versions account for differences in characteristics. That there must be some sort of purple factor that makes this plant purple, and there must be some sort of white factor that makes this plant white. So he was calling these things factors, but what he essentially was getting at was genes. But he just didn't know what genes were because nobody knew what DNA was yet at that time when he was doing his work. So that's one hypothesis, is that essentially there are genes that affect what color a flower is. Well, what we'll be calling these throughout the rest of the chapter is alleles, right? So there's a purple allele and a white allele. So if a plant has a purple allele, it'll be purple. If it has a white allele, it's going to be white. The next hypothesis that Mendel came up with gets at parentage and gets at heredity. So he surmised that for each characteristic, say flower color, he surmised that for each one of those characteristics, a living thing gets one allele from each parent. So it essentially gets one factor from each parent, right? So it's a little bit from mom and a little bit from dad. And we now know this is the case because of chromosomes, right? So you get 23 chromosomes from mom, 23 chromosomes from dad, right? Those 23 chromosomes from mom, those have genes and alleles on them, and the 23 from dad have the same. They have genes and alleles from dad. So we get alleles from each parent. And so then there's some possibilities because you have two alleles, one from each parent. You can be what's known as homozygous, meaning that for a particular trait you have identical alleles. So for instance, a pea plant might have two copies of the purple allele, in which case it would be homozygous, right? So the prefix homo referring to things being the same, zygous referring to chromosomes and things of that nature. So having two identical alleles is homozygous. So you can probably guess what we would call it when you have two different alleles. That's called heterozygous, right? So the prefix hetero meaning different. If you have two different alleles, then you're heterozygous. So for instance, if a pea plant has one purple allele and one white allele, it would be heterozygous. Okay, and then so then what kind of explains what's going on here with these plants gets at Mendel's third hypothesis. And the third hypothesis has to do with what goes on with heterozygous individuals. So Mendel surmised that not all alleles are created equal, right? So some alleles are dominant and some alleles are recessive, meaning that if you put together a dominant and a recessive allele, the dominant allele dominates the situation, and the recessive allele has no effect. It is essentially null and void. And that's what's going on here, right? So this plant right here, we know that it got a purple allele from one parent and a white allele from another parent. So it is heterozygous. It has one purple and one white allele, but it is purple. And so that should then tell you which of the alleles are dominant and which allele is recessive. So if you think that the purple allele is dominant, you are correct. Because in this particular case, the purple allele is dominating the white allele, which is recessive. So not all alleles are created equal. Some are dominant, some are recessive. And with heterozygous individuals, you see the effect of the dominant allele. So the dominant allele dominates the situation. So that was Mendel's third hypothesis. And then with the last one, we're going to kind of start to get at what is going on down there. And Mendel, through all of his work in creating, gallons and gallons of pea soup, he figured out what was dominant and recessive for each of these different traits, right? So the purple flower color is dominant to white. In yellow seeds is dominant to green. In round seeds is dominant to wrinkled seeds. So he figured out all of that. Okay, so then Mendel's fourth hypothesis kind of gets at what you learned during meiosis. So you learned that in meiosis, eggs and sperm are being created, right? Because it's gametic cells are made during meiosis. And each sperm or egg only carries one allele for each characteristic. And that's because you only put half of your chromosomes into an egg or sperm. Right? We learned about how as adults we are all diploid. We have two sets of chromosomes giving us 46 total, but the eggs and the sperm that we create only have 23 total. They are haploid. And the result of that is that you would only put, say, this chromosome or this chromosome into an egg or sperm. So you're only giving one of the alleles to your offspring, essentially, right? So the alleles segregate from each other, which is why this is called the law of segregation, right? So you don't give two alleles to your offspring, you give just one allele to your offspring. So let's talk a little bit about Punnett squares, and then we're going to go back to Mendel's result and kind of visualize it within what's known as a Punnett square. So if you're rolling your eyes because you've already done Punnett squares before and you think they're really boring, that's fine. Just skip the video ahead. But if you're not familiar with them, let's go into these a little bit more and talk about what they're used for. So what Punnett squares are used for is showing combinations of gametes that could occur when you put together two individuals, right? So what are the possible combos of eggs or sperm that could go together when two individuals reproduce? And what happens is that we're going to make these squares, and each square essentially is kind of like it's like a bingo card. Each square represents one possible outcome of fertilization, so one possible outcome of eggs and sperm being put with each other. So let's talk about how monohybrid crosses work, and we'll use and we'll use some Punnett squares to figure that out. So first we're going to look at what goes on here, then we're going to look at what's going on here, and then finally we'll look at what's going on here. Okay, so let's talk about how that monohybrid cross worked. And we'll use so we'll use some Punnett squares in order to figure out what was going on there. All right, so first we'll figure out what was going on with the parents, and then we'll talk about the grandkids at the very end. So we started off with two hybrid plants, right? So we had a hybrid purple plant. I'm sorry, we had a purebred purple plant, and we had a purebred white plant. And you'll see in a minute why I'm writing that in there and there. So we're going to use letters in order to indicate alleles. And so remember, each organism has two alleles, one from each of its parents. And in the letter that we'll use in this case will be the letter P. And so we're going to use capital P for the purple allele, and we'll use lowercase p for the recessive, for the white allele. And that's typically how it's done, where the dominant allele is made capital, and the recessive allele is made lowercase. So in the case of the purple purebred plant up at the top here, the alleles that it would have would be capital P, because we know that it is purebred and it's purple, meaning that it must have two capital P's. The white one down here is going to be lowercase p, because it's purebred, just like the purple one, but it's white, meaning that it must have two copies of that recessive allele, of the lowercase p allele. So what we're then going to do is we're going to see what combinations we can make of the letters that the purple plant has and that the white plant has. And we're going to do that using a Punnett square. So I'll draw in our square like this. So how it's typically done is you have four boxes in the middle, and then open boxes on the top and on the side. And what you do is you take the alleles that each parent has and put them in the top or the side. So we take this capital P and move it right there. We take this capital P, move it there. We take one lowercase p and put it there, and we take another lowercase p and put it right there. And so a question that pretty much every student asks me is, does it matter where you put which letters? And it really doesn't. So as long as you keep the purple plant and keep its alleles together, either on the top or the side, it doesn't matter. So we could have put the purple plant on the side, and we could have put the white plant on the top. We could have switched these two purple alleles and had that one go there and this one go over there. It really doesn't matter. So then what we're going to do next is we're going to fill in the four boxes in the middle by taking the letters on the top and the letters on the side. So we're going to take this capital P right here, and we're going to fill it in going downward. Right, so we're going to put in a capital P in that box and a capital P in that box. Then we do the same with this capital P. We're going to fill it in going downwards. And then we do the same with these lowercase p's over here. We fill them in going across. So lowercase p there, one there, and one there. Okay, so inside of this Punnett square, all four of the boxes resulted in capital P, lowercase p. So that, first of all, think to yourself, is that homozygous or heterozygous? If you're thinking heterozygous, you're right. So remember, hetero means different, and so we have two different alleles right there, one capital P and one lowercase p. So we have a heterozygous offspring. And let's think, is this heterozygous offspring, is it purple or is it white? So we have to remember back to which is the dominant allele. Is it the purple allele or the white allele? In this case, it's purple. So all of these offspring, so each one of these boxes right here that represent one potential offspring, in each of those cases, they're going to be purple, right? So this is why we had that outcome. where when we cross a purebred purple plant with a purebred white plant, all of the offspring end up being purple. So, but then things got more complicated when we started to mix these individuals with each other, when we cross those heterozygous individuals with each other. So let's then do this on the right here. So we have one purple offspring that is capital P, lowercase p. And then we have another purple offspring that is, again, capital P, lowercase p. So we're gonna breed these two individuals together, these two heterozygous purple flowered plants, and see what their offspring look like. So we're gonna create a similar Punnett square. And again, we're going to fill in the alleles that the parents have. So we're going to put that right there, put that lowercase p right there. Same thing over here. So a capital P there, and then a lowercase p there. And again, we're going to fill in the boxes going down. So capital P, fill in the boxes going down, lowercase p, and then fill in the boxes going across. So capital P, and then lowercase p, p. Okay. So so we ended up with one individual that's capital P. Right? So this right here. Then we have two there and there that are capital P, lowercase p. And it doesn't matter, so this we add lowercase p first, but like I was saying before, the order doesn't really matter that much. So capital P, lowercase p, we've got two of those. And then finally we have one that is lowercase p. Right, so this one right there. And so each of these four boxes, as I was saying before, each of the four boxes inside of a Punnett square represent an equally likely outcome when the two parents get together and make offspring. So essentially this first box here, box number 1, 25% chance of that happening. Box number 2, 25% chance of that happening. Box number 3, 25%. Box number 4, also 25%. So we can kind of count up the boxes and figure out what we would expect amongst the offspring. Right, so we have capital P, that's going to be purple. Capital P, lowercase p, still purple. And lowercase p, white. Okay, and we only had one, so 3 out of the 4 boxes ended up being purple. 1 out of the 4 boxes ended up being white. So that's where we get this ratio of 3 purple to 1 white. That's why we have that 3-to-1 ratio of the offspring. Is because that for every offspring that's created from those two heterozygous individuals, from this parent here and this parent here, for every offspring that they have, on average, we would expect a 75% chance of it being purple and a 25% chance of it being white, or a 3-to-1 ratio. So that brings up an important point of all this. And the point is that what's going on here is probability, right? So like I was just saying, there is, in the case of that last cross we did, there's a 75% chance that for each offspring, there's a 75% chance that it is purple, 25% chance that it's white. But these things follow the rules of probability in the same way that flipping coins follows the rules of probability, right? Each one of those parents, could have passed on a purple allele or a white allele, and which gets passed on is essentially a coin flip. And so you know that on average, when you flip a coin, you have a 50% chance of it being heads and a 50% chance of it being tails. But a really important thing is that you don't always get exactly 50-50. So if you were to flip a coin, say 100 times, right, you would expect to get 50 heads and 50 tails. But you might get, say, 60 heads and 40 tails. You might get 70 heads and 40 tails. Or you might even just flip a coin 100 times and get tails every single time, right? It's possible. On average, we would expect it to be 50-50, but you can get all of those different variations of, say, 100 tails and no heads, or like 99 heads and one tails, right? Those are possible outcomes, which is why all of this is based in probability. Same thing as flipping a coin, essentially. So each one of those plants that has one purple and white allele, on average, we would expect it to pass on the purple allele, 50% of the time, and white 50% of the time, but it doesn't always happen. So genetic crosses, they obey the same rules as probabilities, so the same exact thing as tossing coins. And so then when we want to put together like different probabilities of different events, we use the rule of multiplication. And so what the rule of multiplication is it describes how when you have two events that occur separately from each other, the chance of them both happening is the product, meaning the multiplication of the individual probabilities. So for instance, imagine you have two coins, right? So two coins, each coin has a head and a tail. The likelihood of flipping one coin and getting a head is a 1 in 2 chance, right? A 50% chance. The chances of then flipping that second coin and getting heads is still a 1 in 2 chance, right? So the second coin is also 1 in 2. But the chance of flipping two coins and getting heads on both is the multiplication of the two separate events. So a 1 in 2 chance times a 1 in 2 chance is a 1 in a quarter chance. So when you flip a coin twice, there's a 25% chance that you'll get heads both times. We can make this even more complicated. Let's say we're talking about flipping a coin 3 times, right? So each one of the times you flip it, it's a 1 in 2 chance. So 1 in 2 times 1 in 2 times 1 in 2 is 1 in 8. So if you flip a coin 3 times, there is a 1 in 8 chance that you would flip heads every single time. Or say you flip a coin 4 times, right? It would be 1 in 2 times 1 in 2 times 1 in 2 times 1 in 2, which is a 1 in 16 chance. So if you flip a coin 4 times, you have a 1 in 16 chance of getting heads every single time. But the important thing is that if you flip a coin, 4 times, you could get all heads, you could get all tails, you could get 2 heads and 2 tails. There's all these different things that could occur. But the actual probability of getting all heads would be a 1 in 16 chance, which then tells us a little bit about genders of siblings, right? So if you were to have 4 kids, the chances of them all being, say, boys is the same chance as flipping a coin 4 times and getting all heads. And that's exactly what happened with my parents, right? So my parents had 4 kids and all of us just happened to be boys, right? So first coin flip, that was me. So then second coin flip, my brother Stu. Another coin flip, my brother Craig. Another coin flip, my brother Larry. I'm assuming my parents wanted at least one girl, but it just didn't happen for them. And so the chances of a couple having 4 kids and them all being boys is a 1 in 16 chance. The same exact likelihood of flipping a coin 4 times and getting all heads. So try to keep in mind that what we're talking about here is probability. So when we say that there's a 3-to-1 ratio of purple flowers to white flowers, like we were talking about before, what we're talking about is a 3-to-1 chance, essentially. You could end up with all white flowers, offspring. You could end up with all purple flowers. But on average, we would expect a 3-to-1 ratio. Okay, so that's all I've got to say about the first part of the genetics chapter. So in the second part, we're going to get into some more complicated forms of genetics because what we've talked about up until now has been pretty simple. So we're going to get a little bit complex and we're going to go beyond what Mendel has taught us.